the data using a Hill model which indicates positivity, positive cooperativity between the binding sites. And okay, we also tested, this is again a single-stranded substrate, and we also tested a structured substrate, this time without a 3' overhang. And in this case, the N-pin domain is essential for RNA binding. So the deletion of the pin domain virtually abolishes the binding. Okay, so to connect functional and structural data, we analyzed the structures of some homologs in complex. The 3' overhang, the pin domain is essential for RNA binding, and we also observed positive cooperativity between the binding sites. So we propose that RNA interacts, the 3' segment interacts with the exonucleolytic domain as observed in the structures, but the upstream RNA segment towards the 5' would bind to the pin domain. And the pin domain is connected with the CSD1 domain by a flexible linker, shown in orange there. So we propose that reorientation of the pin domain may be associated to a mechanism to direct the RNA substrate to either the top or the side channel, depending on the nature of the substrate. And if we remember our intermediates, approximately 11 nucleotides is the distance from the 3' end, which is the catalytic site, to the entry of both channels. And it's also the size of the overhang, which is the intermediate resistant to degradation. And also 7 nucleotides is the distance between the catalytic site and a 3' helix region, which is involved in RNA unwinding in bacterial RNAs are. So we propose that in the case of a structured substrate, the stem loop hinders the enzyme activity until eventually it reaches the 3' helix region where unwinding may occur. So the flexibility between the domains is important for RP44 activity. We are now trying to co-crystallize the enzyme with longer substrates to try to see the interaction with the pin domain. And to complement our structural and biochemical analysis, we wanted to investigate the effects of downregulation of RP44 in the brucei cells. So for this, we applied the RNA interference technique to generate the brucei knockdown cells. So it's a genetic system which allows to downregulate a protein in the brucei cell, so we can analyze the effect of the depletion of the protein. And also in parallel to rescue the RP knockdown, we have used synthetic genes variants, which are the same amino acid sequence of TB-RP44, catalytic site mutants, and the RP44 devoid of the pin domain, the same construction we have crystallized. And using this regulated complementation system, depletion of the endogenous RP44 can be synchronized with induction of RP44 substitutes. So we can analyze the effect of the mutants. So here I show the cell proliferation curves. In black, we see the control cells, so cells, the growth curve, and in dashed and colored colors, in colored lines, the RNA interference and complementation cultures. So the three interesting observations here are that RP44 is essential for the brucei cell proliferation, since the knockdown cells stop. Induced RP44 is able to rescue the growth phenotype of control cells, and interesting inactivation of the exonucleolytic sites, here by mutation of aspartic 511, and deletion of the pin domain are lethal, but not the mutation of the endonucleolytic site. This is in accordance with our previous results showing that the catalytic activity of RP44 is essential, exonucleolytic, and the pin domain has a role in RNA binding. And to analyze the, I have some time? Yeah. Okay. And to analyze these cellular effects, we applied cryo soft X-ray tomography. So this is a very nice technique to study cells, and it's an image technique that allows visualization and quantification of volumes and surface in near native conditions. So we use synchrotron radiation in the soft X-ray range, known as the water window, between the K absorption edges of carbon and oxygen. And in these energies, the linear absorption coefficient of organic material is about one order of magnitude higher than that of water. And so the chemical composition of the biological structure provides sufficient contrast for the incident identification. So phosphate content, and they play an important role in cell homeostasis and calcium signaling. And we did spectral microscopy, so by collecting data at the pre-edge and calcium absorption edges and subtracting the images, we can specifically identify and quantify, as I show in the third image in red, the organelles which are rich in calcium. So we observed an increase in number and volume of also the calciosomes in the knockdown cells. And a final point we investigated is the autophagy response. Autophagy is a cell response to damage. So it's a cell mechanism to remove dysfunctional parts as molecular organelles. And we used an autophagosome marker, which is a protein which is directed to autophagosomes, fused to a green fluorescence protein. So by fluorescence microscopy, we can quantify the autophagosomes. And we compared the TB-RP44 knockdown cells with cells grow under nutritional stress, because we know that starvation causes autophagy. And again, we observed that in knockdown cells we have a higher number of autophagosomes, so depletion of RP44 induces autophagy response. To summarize, using multiple techniques, our structure and biochemical analysis of T. brucei RP44 allowed to describe at atomic level the endo and exonuclease domains. Our results indicated that the catalytic activity is most exonucleolytic, but the PIN domain is important for RNA binding. And we proposed a model for RNA binding and degradation. The cellular analysis, so we showed that depletion of RP44 in T. brucei cells. I didn't show here, but we have also other results showing accumulation of unprocessed ribosomal RNA and other types of RNA. But depletion of our RP44 causes multiple cellular alterations and inhibition of cell proliferation. So I'm finishing by thanking the people involved in this work, mainly in particular Giovanna Serro, my former PhD, who did most of the work I presented today, a postdoc in the lab, Eloise Lompo, my collaborators from Carlos Chagas Institute, Synchrotron Soleil, and also Synchrotron Alba, the funding agencies. And
thank you for this very entertaining and delighting talk. Being a metallogenic chemist, I know that in most enzymes, metals playing an important role. So I spotted 2 manganese and 1 zinc in your structures. Is this known to be just counterbalance the ions, or is there some proton-coupled electron transfer known, or is this intriguing to change the structure while the oxidation state of the manganese is changing, or could you comment on that?
Yes. Thank you for the question. In fact, the 2 manganese and the 2 magnesium that we see in the endonucleolytic side and the exonucleolytic side, I know it's described the 2 metal mechanisms of catalysis, the transfer electron for the nucleophilic attack for breaking the phosphodiester bond. And the zinc, I think it's structural. The zinc we observed in the endonucleolytic site is new, but I think it's structural, and it's probably related to RNA binding, because several RNA binding domains bind zinc, and maybe...
There's no doubt about oxidation state 2.
Yes.
But manganese may change.
Yes. And it's not clear, for example, because in the case of the catalytic mechanism, you can have either magnesium or manganese, but some enzymes are most active in the presence of magnesium, and some in the presence of manganese. And we cannot explain that in the level of the structure, but we see in the gradation assays that for some, and the catalytic site is very conservative, so that there is no apparent reason, but
at that level of resolution, it should be possible to distinguish between manganese and
yes.
manganese.
With the anomalous map, we could distinguish, because at the beginning we didn't know if it, and then we collected data at the absorption match of manganese, and then we knew that there was manganese at the endonucleolytic site and magnesium at the exonucleolytic site.
Thanks very much.
Thank you.
Thank you for the talk. I'm just curious, is there anything related to your project with other related pathogens like Leishmania?
Yes, I have some targets from Leishmania. Mostly, I didn't work with the RP44 from Leishmania. The reason we chose Trypanosoma brucei is because of the RNA interference mechanism. So we could test the cellular effects and see if the enzyme is essential, because it's also important for a target. And we don't have this genetic mechanism in Trypanosoma cruzi and Leishmania, and then that's why we focus on the brucei enzyme. And so I don't have results on Leishmania homologs. And we have another project, but it's most for initiation factors, translation initiation factors, which we are also investigating Leishmania targets. But they are pretty conservative, so we hope that our results in Trypanosoma can be...
I'm just thinking maybe if you get some inhibitors for this, they can also work for Leishmania.
Yes.
Exciting. Is there anybody else? Any other questions?
If not, I think we should say a very big thank you to Beatriz for a wonderful work. And then we bring this session to an end. I think we're off to an amazing start for the conference. Don't you agree with two outstanding speakers? Before I say anything else, can I just ask you to thank both Emmanuel and Beatriz one last time. And that brings us to the end of the morning session. You'll notice in the program that we've given a very generous lunch break. We don't think it's because the queue at the food will take so long, but we're hoping that it will encourage people to perhaps get something to eat, and then meet up with a friend or two, perhaps a new friend that you haven't met yet, and go and explore some parts of the BOMAS compound that we're so lucky to be in for this week. But please don't lose track of the time entirely. We have a wonderful program for this afternoon as well, and we have the session will start at 2 p.m. So look forward to seeing you all there. Thank you.
One moment, please. Excuse me, one moment.